class. So the topic that we're going to discuss today is about central dogma of molecular biology, also called as central dogma of molecular genetics. So central dogma class, so this is a process, or this is actually a flow by which proteins are made from DNA replication. We have DNA replication, or the duplication of DNA, to translation and transcription. Okay, so to understand the process of making proteins, so we first need to understand the DNA structure. So it's again, class in central dogma. So these are the topics that I include. So we have the DNA structure, the DNA replication, transcription, and translation. Okay, so all right. So again, to understand central dogma, we need first to understand the DNA structure. Okay. If Gara is present, just give me one moment.
Present, sir.
Okay. All right. So the DNA structure class, so its shape is a double helix, according to Watson and Crick. So they are the ones who proposed this structure of a DNA, which is double helix, and the DNA. Each backbone of DNA runs in an opposite direction. So here, so this one class, okay, so take a look at my okay, so this one class, okay, so this is what we call the phosphate, or the backbone of the DNA. So this, the first strand runs in this direction, so from 5N to 3N, and the other strand runs in an opposite direction from 3N to 5N. So it goes this way for the first strand, and the second strand goes that way. Okay, that is why your DNA runs in an opposite direction. Kindly unmute your microphone, please. Kindly mute your microphone, please. Okay, thank you so much. All right. So, mauna siya again, class, ang structure of the DNA according to Watson and Crick. So, the backbone of the strand consists of sugar, or the sugar called deoxyribose, and a phosphate group. So, if we are going to take a look at this structure, so this is the backbone of the DNA. So this is composed of the phosphate backbone again, and the deoxyribose sugar. So if we are going to zoom in nato siya, so we will see the chemical composition of your DNA. So this one is the phosphate group, and the deoxyribose sugar. So this serves as the backbone of DNA. Again, class. So DNA's backbone is composed of the phosphate group and the deoxyribose sugar. That is why the DNA is the DNA, or how do I say this one? Like a meaning sa DNA is the deoxyribose sugar. Yeah, in deoxyribonucleic acid, because again it is composed of phosphate, deoxyribose sugar, and nucleic acids, or the nitrogen bases. Okay, so the ranks of the helix class. Okay, so the ranks kaning murag mga hagdanan. Okay, nga nagababag. Okay, those are your nitrogen bases, okay, or N bases, and there are 4 types. I think you're already acquainted with the terms with nitrogen base, by the way, since during the first day of our discussion, I have already told you about these. So there are 4 types. We have the adenine, thymine, guanine, and cytosine. And in RNA class, there is a uracil, but uracil cannot be found in DNA, only in RNA. Okay, so let's focus on this four first. So we have adenine, thymine, guanine, and cytosine, because this is the four nitrogen bases or the N bases of your DNA. Okay, and one set of this. Okay, so one set of a phosphate backbone class. Phosphate backbone, we have your deoxyribose sugar and your nitrogen bases. So this one set of this is called your nucleotides. Okay, and this one another. Okay, so this is another set. So this is also called as a nucleotide. This one is also another set. This one, and this one. Okay, and your nitrogen bases is classified into two types. We have your purines and pyrimidines. For purines, nitrogen bases that are classified as purines, we have your guanine and adenine. Okay, they are classified as purines because that is due to their chemical composition. So adenine and guanine are two ringed, while your thymine and cytosine, they are called pyrimidines because since both are one ring only. So if you are going to take a look at their tanawa. So si adenine murag duha iyang ring, then si thymine kay usara. Same po si guanine duha, cytosine usara. Kaunang si adenine ug si guanine are classified as again purines, and thymine and cytosine are your pyrimidines. Okay. And according to Chargaff's rule, so N bases have a base pair. So nasa sila partner, and each N bases has their own partner, or what we call this one, or base pair. Okay, so si adenine double bonds with thymine, and cytosine triple bonds with guanine. Okay, that is always the case. Una siya permiklasa ng remember, si thymine, the only partner or the only base pair of thymine is adenine, and vice versa, and they are connected via a double bond. Cytosine's only partner is guanine, and vice versa, and they're connected via a triple hydrogen bond. So dili pwede nga si cytosine mo partner ni adenine kay multiple bond si cytosine, then si adenine pang double bond. So it is not possible. So it is always thymine, adenine, cytosine, and guanine. Okay. And for their connections class, so your phosphate group, deoxyribose, and N base and your nitrogen bases. Again, so the connection between phosphate group, the sugar, and the nitrogen base are strong covalent bonds. So kini siya, kini, if i-connect na sila nga duha, that is, so they are connected by a strong covalent bond. But the connection between your two nitrogen bases, so kini connection sa thymine and the connection of adenine, are a weak hydrogen bond. Okay, that is why it is easily the helicase enzyme can easily break this bonds during replication process. Okay, right. So this is, by the way, the timeline of a genetic study, starting from 1866 with Gore Mendel, that publishes his first work on heredity of meat. So Mendel is basically the founder of the study of the genetics study, and up until this timeline, it's only up until 2013, in which Crick's 1953 letter to his son describing double helix. The double helix shape of a DNA costs around $5.3 million. So yeah, familiarize the timeline class. Si Elcano is present. Okay. So do you have any questions regarding the structure of DNA? Do you have any question, class? Kahilom ba ninyo? Wala? Okay, I think silence means no, wala may question. Okay, so let's proceed to the DNA replication class. So the basic definition of DNA replication. So this is the biological process of producing two identical replicas of DNA from one original DNA molecule. So this process, the replication process class, so if the DNA replication actually happens during the interphase. So cell division, so the DNA replication happens on the interphase stage of your cell cycle. So before we proceed further, I want you to watch a video. Okay. Stop sa share ha. Class, nakita mo sa akong screen?
Yes, sir.
Okay, wait ha. Makadungog ra mo sa audio.
No, sir.
No? Ay, share ko na lang sa ano. Ay, so, sige. Okay, I will share this one na lang sa ato ang chat box. Then, okay class, I'll give you 5 minutes to watch the video and then proceed ka sa atong discussion. Okay, di man ko katungog sa audio and I don't really know how to make you hear the audio, so sige na lang. Sige, I'll give you 5 minutes to watch the video. That is about DNA replication. So that video is very detailed, by the way. So that that video you can see all of the enzymes that is responsible in replicating your DNA. Okay, so I'll give you 5 minutes and let's continue at around 9:40. is the unzipping of the double helix structure, and it is separated by the enzyme helicase. Okay, so helicase is the unzipping enzyme. So that enzyme is the one who breaks the hydrogen bonds between complementary bases. Okay, and once the helicase enzyme unzips the DNA structure or the double helix structure of a DNA, it will form a replication form, which provides a template for creating a new strand of DNA. So once na-unzip na ni helicase ang double helix structure of the DNA, these kanang mga unzip na DNA class are called your replication form and will serve as a template for that new DNA. Okay, so next is that. So an enzyme called primase makes a small piece of RNA called primer. So the primase class so the enzyme called primase will attach to the replication fork and makes a primer. So the primer serves as the starting point in creating strands of DNA. So if wala ganay primer class ang replication fork sa DNA, so your DNA polymerase enzyme will not be able to make new strands of DNA. That is why a primase enzyme is essential, and of course, creating primers so that your DNA polymerase enzyme can make new strands of DNA. Okay, so remember the enzymes we mentioned. So we have the helicase that for helicase unzips, forms a replication fork. Then primase will attach to the replication fork and will make small pieces of RNAs called primers. Okay. All right. Next, we have the enzyme called the DNA polymerase binds to the primer, okay, and will make new strands of DNA. So katong si primer ganina, for example, natin si primer katong enzyme na primase. So your DNA polymerase will attach to the primer and will start making new strands of DNA. So sa DNA class, so your replication fork sa DNA class, so the unzipped DNA structure again is called your replication fork, right? So your replication fork, so na the high leading strand and lagging strand. So the leading strand runs from 3 to 5 from 5 to 3 direction, and your lagging strand runs from 3 to 5 direction. So in your leading strand class, the DNA is made continuously by your DNA polymerase. And in the lagging strand, since it runs in an opposite direction, your DNA polymerase cannot add N bases continuously, so it needs several primers or RNA primers in order for DNA polymerase to add 2 N bases to the lagging strand. And the DNA polymerase will only make small chunks of DNA, and that is called your Okazaki fragments. Okay, remember this term, the Okazaki fragments, the enzyme DNA polymerase, leading strand, lagging strand. And your enzyme and exonuclease class, they are the one. Okay, so the enzyme exonuclease removes the primer from both strands. So primer sa leading strand, and there are also several primers in the lagging strand. So the enzyme exonuclease removes those primers, and then your DNA polymerase again fills in the gap that was nabilin sa mga primer. Okay, sa mga primer, because once primers will be removed since they are RNAs man. So na mga gaps na diha sa, most especially sa lagging strands. So your DNA polymerases goes into picture, and of course will fill the gaps with nitrogen, with the complementary nitrogen bases or N bases. Okay, and lastly, the enzyme ligase will seal up both strands of DNA to form a continuous strand. So it is like the ligase kanang aids in hydrogen para kato mo perform og hydrogen bond katong duha ka N bases, like for example si adenine and si thymine. So ligases, it's like they are trying to glue both strands of DNA in order to form a double strand of DNA, both sa leading strand and the lagging strand. Okay. So, and according to the video that you watched earlier, so the DNA replication is described as semi-conservative. It is because your DNA is actually made up of new strands of DNA and an old strand. So here in this diagram that we have in our presentation. So the blue ones are the old strands of DNA and the new ones. So the new ones are colored in red. So these are the strands that was made by your DNA polymerases, and that is why, and the DNA replication is described as semi-conservative. That is because your DNA, the new DNA is composed of the new strand and the old strand. Okay. And of course, class mistakes during the DNA replication process is removed and repaired, or that is what we call proofreading by your DNA polymerase enzyme. So our DNA polymerase enzyme checks for damages, mistakes, and once the DNA polymerase is able to find one, you will replace the damaged DNA, or will repair the damaged DNA. So that is also the job of the DNA polymerase enzyme. Okay. So do you have any questions, class? DNA replication. So far. Okay, so if you don't have any questions for DNA replication, so let's proceed to translation. So, okay, class. So this is the summary of the central dogma of genetics. So we have the DNA. So we have the DNA. So we have your transcription. The basic definition of transcription is just making mRNA strands out of the DNA strands, and of course the translation is making proteins from the RNA code. So mauna siya ang explanation sa central dogma class. So from DNA to RNA and protein or polypeptide. Okay. So in the transcription process, class, so that is making mRNA from DNA. So the enzyme called RNA polymerase, so hindi kayo siya kitanon sa atom diagram, but there is a pink square over here, so that represents the RNA polymerase enzyme. That enzyme will attach to the start of the gene and makes a strand of messenger RNA or mRNA. So kanisiya class, so here kaning pink na color dere around the start of the gene is your RNA polymerase. So attach na siya sa gene, and then if that RNA polymerase enzyme will create messenger RNA. So muna siya ang messenger RNA class that was created by created by the RNA polymerase. Okay. So the DNA template for transcription is 3N to 5N. So your mRNA was created from the DNA, from the sense strand of the DNA, which runs from 3N to 5N. That is because your RNA will always run from 5N to 3N. That is why your mRNA codes are created from the 3 to 5 end of the DNA, called the coding strand or the sense strand. Okay. So in this figure, as you can see, so this is the DNA coding strand class. So mauni siya ang mga N bases, mga combination sa N bases. So if nanagani is mRNA, mauni siya ang iyang mga complementary N bases. So we have sa DNA coding strand, if we have BCG, DAG, CAG. So its mRNA codons would be the AGC. So AGC, since opposite of thymine is adenine. We have C, opposite of the pair of your cytosine is guanine. Guanine for cytosine, thymine for adenine. And here, class, as you have observed, you cannot see an N base. You cannot see thymine in mRNA codons, and it is replaced by your uracil. Okay, so ang partner na ani sa adenine class sa mRNA would be uracil, so not thymine. Okay, basta mRNA or any type of RNA class, you cannot find any thymine in there, and your thymine is replaced by the N base called uracil. Okay. So partners sa adenine will be uracil in mRNAs, and of course CAT, that's GUA, that's guanine, uracil, and adenine. So muna siya ang complementary strand. So the mRNA codes was taken from the DNA codes that we have here. Okay. Since maumaning DNA codes, muna siya, this will be the RNA codes. So I think I have included this one in. I used to include this one in our assessment last P1, no? Atong first na itong assessment. Ako'y question nga inaing ani sa ato ang assessment first. Atong assessment pag first. So, sa mRNA again, sa mRNA, mauni siya ang complementary codes from the DNA coding strand. Okay, so mauna na siyang transcription process class. So yung transcription process is just making mRNA out of DNA strand. So after that class, so there is some sort of deletion process before the mRNA will go out to the nucleus of the cell and proceed to cytoplasm for the translation process. So the mRNA has introns on the strand. So ang buhaton ana is mag-RNA splicing rod, and the introns will be removed para mahimong siya in-aning nga strand. So naman ay mga introns or mga molecules that is not needed for the translation of the DNA, so it will be removed through RNA splicing. Na siya para mahimo siyang in-ani, or para ang atong mRNA strand will be complete, and before it will proceed to before it will go out to the nucleus of the cell and goes to the ribosome for the translation process. Okay. So for the translation class, once na ng mRNA, so your mRNA will go out to the nucleus of the cell towards the cytoplasm, and that mRNA will attach to the ribosome class, and the ribosome your ribosome will read the mRNA strands and will produce the amino acid sequence or your protein. So kani mRNA strand. So for example, kani ang atong mRNA strand. So this mRNA strand will go to the ribosome, then i-code na siya sa atong, and i-code na siya kanang mRNA strand sa atong tRNA which is inside the ribosome. So if inani a strand sa mRNA class, so the strand that will be ang strand sa atong ang code sa atong tRNA will be this. It is still an opposite or like the opposite for the mRNA. So the opposite of A is U, opposite of G is C, opposite of C is G, and so on and so forth. And then each tRNA codes has an attached amino acid, has or contains an amino acid. So these amino acid, once ma-encode na siya sa RNA, will join together, will join us together until until there are enough amino acids that can produce a protein. So for example, kami siya ang mRNA code. So if inani ka ng mRNA code class AGC, so pangitaw na to siya kung unsa nga protein or nga amino acid ang ma-produce sa AGC nga combination, and that is the amino acid called serine. AUC, that's isoleucine, and your GUA is your histidine. Okay. So mukode na nga mukode si RNA class until and mo-stop na si tRNA code once na stop codon appear sa tRNA. So ano siya ang mga possible combination class sa mga N-bases class na maka-create of protein. So like for example, U here. So kung sa mRNA, U ni siya diri, for example, so ang iyang combination will be your PHA, that is phenylalanine. Okay. Mo siya possible combination class. And if magtanaw gani mo sa possible kung unsa nga protein ang mahimo ang mabuhat sa inani nga combination class, always tanawon ninyo permi kung unsa ang combination sa mRNA, tagtulo-tulo na siya class. So if inani gani ang combination sa tRNA, ang protein will be, for example, AGC, ang amino acid will be the serine, AUC, isoleucine, and your GUA will be your histidine. Okay? So mao ni siyang mga lists sa amino acids class nga sa ribosome. Then these are the list of possible possible codons na makita nato sa atong mRNA. And each combination kay nasay own nasay mga amino acid, nasay respective amino acid. Like again, for example, UU for phenylalanine, UUA for leucine also for CUU, CUC, CUA, CUG, and so on and so forth. Okay, like for example, if inani ang DNA strand class sa transcription process, so this is the DNA strand. So mo sulod si RNA polymerase enzyme and will make an mRNA strand nga ingon ani. So ingon ani mo gawa sa mRNA strand. And then sa tRNA, kung mag-code na, inani ang mugawa sa tRNA class, okay, it is just the opposite of the mRNA. And for the amino acid sequence, we have your AUGs for methionine, CUG for leucine, GGC for glycine, CUG for leucine, UGC for CYS for cysteine, arginine for AAU, alanine for GCU, GCA is arginine, UCA is serine, GUC is valine, and UAA as a stop codon. Once na nagani stop codon nga mo appear class mo undang si tRNA o code na o protein or code na o code sa mRNA. Okay? Okay, so oras na siya class. So do you have any question? May question, class? Clarifications? Okay, so this PowerPoint, I will be sending this PowerPoint presentation later on, or I will post this PowerPoint presentation later on sa ating classroom. Kahilog ba? May question, class? Or wala na?
Sir, excuse me, sir.
Yes?
Bali, ang tRNA, sir, is basically just DNA, pero ang thymine is gihimo ra siya giracell.
Yes. Mura na siyang DNA strand siya, but the kanang thymine gihimo ra uracil. Correct. Sakap.
Okay, thank you.
Okay, any other question? Okay, kung wala na, di ba, nag-ingon man ko last week na last Saturday nga mag-assessment aron sa. So, sa atong assessment class, what time niyo gusto ganahan mag-take sa assessment? Kana na lang din.
Hello, sir. Pwede afternoon, sir?
Oo, sa oras. What time niyo gusto?
Sir, sa assessment, sali na ba itong new?
Oh, ayan. Yes, ako i-appeal ang translation of transcription sa assessment. So, mas maayo dyan yung afternoon mag-assessment mo para makastudy pa mo sa bago na itong lesson. So, mga what time niyo gusto? So, afternoon, okay ko sa afternoon. So, what time niyo gusto? 3 p.m.?
Pwede 3 p.m., sir?
3 p.m.? 3 p.m. niyo gusto? Alas 3? Sige daw, kinsay ka na 3 p.m.? 3 p.m. or 1 p.m.?
3 p.m. 3 p.m., sir?
Sige, 3 p.m. na lang. Sige, mag-assessment na ninyo later ha, 3 p.m. Okay? So, okay, para syempre maka-study, maka-study po mo.
Thank you, sir. Sir,
Yes?
about sa module 15, sir, wala po kaming hard copy.
Module, don't worry, I will send again a book soft copy ninyo.
Okay, but thank you.
Okay. So, maroon itong
Module
assessment.
mag-start, sir.
Ah,
Module mag-start ang assessment.
what time mag-start?
What module, sir?
Okay, so, okay. Ang sa topic na itong Thursday. Okay, sex-related inheritance. Kato, mga sex-related inheritance such as your X-linked trait, or sex-linked trait, your sex-limited trait, and sex-influenced trait. Mag-start na dito. Then, I will also include the topics that we discussed earlier. Itong DNA transcription, translation, replication. Just ka lang, class, remember kung unsang, di ba nairato ninyo mga enzyme sa DNA DNA replication kay magnawas dito na sa akong assessment later. Okay, module number, module number 14, 15, and 16. Okay?
Okay, sir. Thank you.
Sige, mag-send ko sa module after Annie. So, napaba ko yung question line. Oh, ako di ay. Sa P1, P2 di ay nato class. Diba, this week marang P2 sa?
Sir. Yes, sir.
So, this week, Friday lang yapon ta. Okay, naamagod may classmate na di ka P2. Saturday. So, ganahan ko nga madungan tanan o take sa P2 ba para tigang ilisod di chikan. So, kaya na lang, mag-Friday lagi akong ta, same time, P1, ha? Para Saturday, makapahuway po niya, magamit po ninyo ang Saturday sa inyong RTE exam or RPE exam, which is scheduled next week. Okay? Okay, class? Understood?
Sir, until what time po ang assessment?
It 2 hours na 2 hours na ako. Anyway, sa'yo naman ang inyong assessment. Multiple choice o majority sa question kay multiple choice. So, ito siya, like 40 items multiple choice ka na. Then ay mga mga problem solving 15 items na so gamay ra ang problem solving so sayon ra inyong assessment later. Ako na naguro na oras 2 hours sa assessment. Okay?
Sir,
Yes.
until what module ang genetics, sir?
Genetics, like the whole
Yes, sir.
the whole module. Okay. There are only 18 modules sa genetics.
Okay, sir. Thank you.
So, unlike sa ubang subjects sa ako systematics na maabot ng 34 modules, yung genetics ka 18 module. So our last topic I discuss tomorrow is about mutation. Okay? Alright, so mauna to siya class if wala na may question. So, ang P2 na to, ako na i-separate ang lab of leg para sa itong P2. Separate na itong lab of leg. So, for example lang niya, matag 1 hour sa lecture, matag sa 1 hour sa lab para or 2 hours na rin, 2 hours sa lecture, 2 hours sa lab para na mo enough time to answer. Okay? So, maura to. So, if wala na mo okay, so if wala na question, kay so, end na nato. Okay, so if wala na may question class, so let's call it a day. I hope you guys will enjoy the rest of the day. Good luck sa assessment later. And stay safe. Okay? See you tomorrow, 2 p.m. 2 p.m. na tomorrow, ha?
Yes, sir. Thank you, sir.
Thank you. Bye-bye.
Thank you, sir.